Hi, hello. Am I audible? Yeah. Okay. So, good morning, good evening, afternoon. Greetings from Project Encephalon. And I am Pranjal Garg, currently serving as the president of Project Encephalon. Project Encephalon is an international trainee-led non-profit organization for neuroscience enthusiasts and conducts various academic and non-academic neuroscience-related activities while accounting for the interdisciplinary needs of the field. It is my pleasure to welcome you all on the first day of Cajal Week, a week-long celebration of Dr. Santiago Ramon Cajal's 169th birth anniversary. Cajal for us is not just a name, it is a spirit. Spirit of hard work, spirit of innovation, spirit of flexibility, spirit of art, spirit of medicine, and most definitely, spirit of neuroscience. This event includes 7 talks, a panel discussion titled Bench to Bedside, Bridging the Gap in Neuroscience, and a social hour with friends and colleagues with engaging activities. I'm really very grateful as well obliged to thank the sponsors of Cajal Week, Scientists Inc. Scientists Inc. is a registered non-profit organization dedicated to science outreach. Amidst this terrible pandemic, I would like to extend my heartfelt and warmest prayers to everyone affected by it. We hope this activity will provide some relief and will keep those busy who are stuck at their homes or in quarantine centers. As human beings, we all depend on compassion and empathy of one another. And I sincerely hope that we all are doing the best we can to help each other. Now, please welcome Anushri to start the event with the keynote address.
Hello, I hope I'm audible. Okay.
Yes.
Thank you so much, Pranjal. Good evening, good morning, or good afternoon again, depending upon where you're joining us from. Once again, a very warm welcome to the opening session of Cajal Week. I am Anushri Krishnamurthy, the Vice President of Project Encephalon, and I will be the moderator for today's session. We will be kicking off this week-long celebration of Santiago Ramon y Cajal's 169th birth anniversary with a keynote address by someone who has been a huge inspiration to me personally, as well as many aspiring neuroscientists of today. She works as a professor at the Swiss Federal Institute of Technology, Lausanne, and leads the Laboratory of Behavioral Genetics that investigates the effects of stress and anxiety on the brain and behavior. She is none other than Dr. Carmen Sandi. Dr. Sandi has many, many feathers in her cap. She is the founder and co-president of the Swiss Stress Network and the co-director of the Swiss National Center of Competence and Research Synapsy. She is also the immediate past president of the Cajal Advanced Neuroscience Training Program and the Federation of European Neuroscience Societies, that is FENS. She is a strong advocate for inclusivity in the field of neuroscience and is the chair of the ALBA Network, a community that works towards diversity and equity in brain sciences. Project Encephalon is also proud to have signed and endorsed the ALBA Declaration on Equity and Inclusion recently. We are beyond elated to have you with us today, Dr. Sandi. I feel there couldn't be a better way to start off with this event than with your keynote address on a topic that has fortunately or unfortunately gained a lot of relevance in the past year. Stress and the individual, the neurobiological mechanisms underlying differential susceptibilities and adaptations. I would like all the attendees to maintain the decorum of this online meeting. You have to put in any of your comments in the chat box and any questions that you would like to be addressed in the Q&A box. I would like to hand it over to you, Dr. Sandi. The floor is all yours.
Thank you so much for this wonderful invitation. I feel very, very honored to be here today. I think I know that it's a very important day for you. It's an amazing project that you have launched. Of course, as soon as I saw the email that you are neuroscience enthusiastic people, I thought, let's share maybe our enthusiasm for neuroscience. And I am really delighted to be with you and to get to know you personally. I think you've put together an amazing program. I will try to follow some of the other talks also throughout the coming days. And I also wanted to join you in this maybe few words thinking of all the people that are suffering also now due to the pandemic. I know that it's particularly bad in India at the moment. It's very difficult times, and I think we can only hope that both science and also our societal maybe approaches and all that we learn, that it's important also to deal with issues related to stress, etc. Maybe eventually can have an impact, but warmly also sympathies to everyone who's suffering. And yeah, I think I prepared something to share with you in two different ways. I think at the beginning I have something more general, also to have a few words regarding Cajal. So I'm going to start sharing my presentation. And let me see here. If there is any problem, you will let me know. Right now, I guess you can see the presentation. Very good. So as Anusri was indicating, I'm going to talk about stress, which is, of course, a very important topic, but also the insights that my lab has been interested in revealing in the last years, how the individual makes a difference in how stress affects us in different aspects. So, of course, we look at factors that are important in stress vulnerability and also try to understand the neurobiological mechanisms that underlie those. But before, I wanted indeed to say a few words regarding this week anniversary, 169 anniversary of Cajal. We have him here probably also in a weekend like we are. Continuing working and maybe looking a bit tired, but also delighted with the work and the discoveries that he was doing. And here we have images that I took from the website of the Cajal Institute, also because I think they represent many different aspects of what he did, his utils, what he drew, etc. But also maybe for those that don't know it, because I did my PhD, in fact, at the Cajal Institute in Madrid. And I wanted to show you here the images. This was the first building where the Cajal was when I did the PhD. Then the building or the institute moved a few meters up in the same street, not too far, but to this building where it's only building for the Cajal Institute, and where I also did postdoc, and I worked afterwards as a senior scientist for a few years before I moved to Switzerland and other countries. But yeah, I wanted to give you these insights, also to share these words from Cajal, that he was amazing because he didn't have the same tools and information that we have nowadays about how the brain works. Still only from images that he saw, he got amazing insights and hypotheses about how the brain worked and how could eventually be plastic. And I think the fact that he already envisioned that the brain can be plastic and that people or ourselves, we can sculpt the brain, I think it's very relevant and also relevant for my talk today, because stress, from my point of view, is one of the major sculptures of the brain. And it's something I wanted to share with you a little bit at the beginning, how I also became an enthusiastic and I got into insights of why maybe stress could be an important sculpture of the brain. I'm not going to have the time to talk about developmental stress so much today because we have 1 hour for the lecture. But of course, from that point of view, it's when one can see that indeed stress can have incredible impact in the neurodevelopmental trajectories of the brain and individuals. But I want to tell you, some aspects of the thinking that brought me to all the insights that we have nowadays, and also the projects that we are doing currently in the lab. I went to Milton Keynes, that's in the UK, to do a postdoc many years ago after the PhD at the Cajal Institute, because they were looking at the neurobiology of learning and trying to understand particularly the molecular signatures of plasticity, synaptic plasticity, and memory formation, long-term memory formation. And the model that they were using was the passive avoidance learning in one-day-old chicks. And basically what this model does is to train chicks to learn what it's edible and what it's not edible, but it's aversive. So these one-day-old chicks, they are interested in anything that it's bright because it could be maybe edible. So when they see a bright bit like the one you see here, they would peck it and that's it. And if it has water, they like it. And if it would have any nutrient as well. But in this model, what it has is an aversive substance so that the animals peck it and they have to learn that this is something not edible. It's in fact something to learn and remember in the future that they should avoid. And this was in the 80s, and indeed there was a lot of work looking into the mechanisms of those memories. And what I realized when I looked at these chicks is that they were highly stressed when they pecked and they were learning. It was not just a cold brain or a cold computer that was processing the information. What they did was reacting very stressed when it was something aversive, moving the head, going backwards, really having a complete stress response. And I thought also because of a background in experimental psychology that perhaps stress could have an integral impact on the formation of the memory, and that perhaps some of the stress mechanisms, like for example, glucocorticoids, that as you know, they are hormones from periphery that can cross the blood-brain barrier, because they are very strong regulators of gene transcription, and we know that protein synthesis is important for long-term memory. I thought that perhaps this stress component could be telling the brain what's meaningful to remember for the future. And that's why instead of continuing with the project that I was supposed to do there, I convinced my supervisor at that time to let me investigate this hypothesis that we showed that indeed glucocorticoids contribute to the strength of the memories that are formed so that eventually we remember better in the future. At that time, and I think maybe this I also want to share with you because I think many times we can have insights for our science that come from our experiences. I was interested indeed now in understanding how stress and the stress-related neuromodulators or hormones could be contributing to the formation of memories. And I wanted, of course, to go also to other models like rodent models, because we were doing this at the Cajal Institute, as you can see here in Madrid. And we were using the Morris water maze. And because I'm very sensitive to cold, like I normally get stressed if it's a bit cold, I thought maybe indeed if it's cold, the animals might be more stressed, and then that might influence the formation of a memory for the hidden platform. I think many of you already know that the Morris water maze is a special memory, and that the animals are trained to find a platform that it's non-visible just by helping themselves with external cues. And what we did was to train the animals at different water temperatures. One was mild, 25 degrees, and the other was cold. And indeed, we saw that those that trained at the colder one, they release higher levels of glucocorticoids when we took blood samples, and they also keep a much stronger long-term memory. And we also saw that the glucocorticoids were causally implicated because we could train them at milder conditions and then inject them with corticosterone immediately afterwards, and this improved the long-term memory. Afterwards, when I already moved my lab here in Switzerland, in Lausanne, we could reveal other mechanisms. And in this case, we modified slightly the protocols, and also we worked with mice. So we changed the temperatures because mice are smaller, and then one cannot use very cold temperatures. But basically, again, we saw that those that are trained at the more stressful conditions, they learn quicker. So we modified the protocol so that the effect was already observed during training, not only for the long-term memory formation. And then we could also implicate the trafficking of AMPA receptors into the synaptic membrane as important mechanisms modulated by stress. So we see that indeed the stress is there shaping how the circuits are working. Also, as I said before, also shaping the long-term remodeling of circuits. And something that started becoming very evident in our studies is that if now we don't change, we don't modify the stress levels, but we look at individual differences, indeed the individual differences were evident, so that even under the same stress conditions, there were animals that were much better to learn than others. And here, these were early times with my collaborator also from Madrid, César Venero. We already saw that if we classify the animals for anxiety, those that are high anxious are normally not so good at learning, particularly at the beginning, revealing that indeed individual differences are critical. And this gave us hints of which type of traits to look if we want to understand individual differences in vulnerability or susceptibility to stress. Something else, and I finish with this first introductory part. What we also did here in Switzerland was to ask if we, instead of using only 2 levels of stress, if we use 3 levels of stress, would we be able to reproduce the well-known inverted U shape response, meaning that both low stress levels and high stress levels are not optimal, and then there would be an intermediate level that it's optimal, because we always hear about this inverted U shape, but when we went to the literature, we realized that people were taking bits and bits of information from different studies, but it had not been demonstrated in a single type of study or protocol. So that's what we could do, and for this we used now not the Morris water maze, but a radial arm water maze. And all that's very good, but because we were already very interested in individual differences, we had pre-characterized the animals' responses to a number of challenges, one related to anxiety, others related to curiosity, exploration, etc. And what was quite interesting to find here is that whereas some of these groups of animals indeed showed the U-shaped response, other animals, like these ones, are not good at all under high stress conditions. They are very good at low stress conditions, and there are other animals that are the opposite, meaning that we are different indeed, and that some of us need additional levels of stress to have proficient performance and others not. And this is something I say we, because I've been asking several times to groups of people, starting with my own laboratory and then afterwards to other audiences. And indeed, people are divided in between those that perform good under high stress and those that perform well under low stress. So all this more related to indeed stress and learning, more when we are talking about acute stress. But something, and I think this is what it's also more relevant for the type of stress that we've been living over the last months and year, is that we know that repeated exposure to stressful conditions can be quite damaging in general for physiology and also for brain and behavior. And this is very relevant to understand the mechanisms, but also some other nuances that have been particularly interesting for us. And it's that indeed, again, here, when we are exposed to adversity, which is more chronic, we are not all equal in the susceptibility that we show. And this is particularly when we look at how people develop a different type of, let's say, depressive-like behaviors. So from stress, if it's repeated, it's normal to get eventually to situations of distress, helplessness, lack of motivation, lack of pleasure, and lack of energy. And that's something that is reflected in these images here that require many times endurance, keep going, and some of the functions that are indicated here, like self-confidence, resistance, active coping, and perseverance. So we know over the years now that indeed individuals, both animals and humans, when we are exposed to a long levels of stress, long high levels of stress, then eventually we lose like the energy. We just give up. We don't have control under the circumstances. But this is, of course, something that doesn't happen equally in all individuals. Some of them have a different threshold that might be quicker or slower. And that's something that it's of high interest because it helps us understanding what could be underlying different susceptibility, meaning both vulnerability and resilience to stress. So we have this situation in which indeed if we examine whatever it is afterwards, but this is quite relevant in the field of stress-induced depression. If we see after exposure to stress, how individuals perform or develop afterwards, we see that not all are equally affected, and that indeed we have some individuals that are quickly and highly affected. And we've been seeing the same in the literature. And this allows us afterwards to to characterize these individuals and then to look for mechanisms, what is different. And this is, I think, very relevant. Eventually, it can help indeed developing treatments, etc. But we've been wondering, indeed, if something is different only in some of the individuals, wouldn't it be that they might be already be different a priori, and that by identifying factors, let's call it biomarkers or other factors a priori, if we know which ones are more vulnerable, we can already start investigating what's different in terms of mechanisms as well, and follow them longitudinally to see perhaps if we can have a bigger impact, either by applying prevention interventions or other treatments afterwards. And also I think because it's quite relevant on its own, because it's what I say, these individuals that are more vulnerable after stress, indeed they develop different symptomatologies, but many times they also have a lower threshold for even lower levels of stress. And that's why we think that it's particularly important to identify who they are and how this vulnerability operates. So I'm going to tell you the type of studies that we are doing now to look at these individual differences, but I want to motivate it a bit instead of just going quickly to the factors that we look into, but more how we got into there. So of course one can do genetics if one wants to identify which individuals are more vulnerable to stress-related disorders or to other psychopathologies in general. But we know after years that this has resulted to be a very difficult task to identify genes that are particularly indicative of different susceptibility, because genetics is complex and genes don't operate in isolation. They interact with the environment and they interact with other genes. And of course, nowadays, the field is moving in a way that I think it makes a lot of sense, which is identifying groups of genes that all put together represent polygenic risk factors. And then eventually the use of these factors or these risk scores can help identifying individuals. But this has been only building up over the recent years. And I think we are also not yet there, and it's quite complex. We undertook a different way, which was to ask if behavior or traits can tell us also something that could be specific regarding the vulnerability to stress. And of course, when we are talking about traits, predispositions, temperament, etc., they are normally factors that put together many different aspects and mechanisms. So probably they might be also reflecting poly mechanisms somehow. So this is a little bit the idea underlying this, but it's using now behavior to see if we can reveal differences in the individuals that are meaningful in this context. So we took large cohorts of rats, and we gave them tests that were of natural exploration to see their natural behaviors when they are exposed to the elevated plasmase. They all have trade-offs between being more protected or being more exposed, or we take out these trade-offs in the circular corridor, for example, so that it's just a corridor with protected parts, and there we can look at just more the explorative, the locomotor aspects, and then for the explorative ones, we introduce a novel object, etc. So different number of tests that the same individuals are run through. And then eventually what we do is to take all the parameters or the meaningful parameters from this test and then apply, in this case, we applied principal component analysis to identify which factors we could extract and how much percentage of variance they could be explaining. And at that time, because this was published already almost 10 years ago, we identified mainly more meaningful factors that we named anxiety, activity, exploration, harm, avoidance, etc. But basically what afterwards we did was to expose these animals to a chronic stress across different weeks. And then we were testing them to see when we look at these factors, is it possible that any of those or a combination of these factors would be predictive of the impact of stress on the depressive-like behaviors that we were exploring. I want to make a long story short here to just show you that when we look at one of these parameters in one of the depressive-like behaviors, that's the forcing test, and we look at percent time immobile, what we see is that across weeks of stress exposure, animals are floating more and more, indicative of a development of more passive coping responses, which are normally more indicative of depressive-like behaviors. And then, yeah, every week it builds up. But then when we look at the individuals, what we identified is that some of these traits that we had identified before, like in this case, it was a combination of anxiety and exploration, but particularly the trait of anxiety explained a lot of the variance that we were seeing, meaning that those animals that are low anxious, after 2 weeks, they don't show differences. And those that are high anxious already, after 2 weeks, they already show differences in this behavior. And then we saw similar, quite drastic differences in this same line with other tests. So indeed, as I said, I am going to just do a long story short. We did many other experiments in which we observed similar results, that whenever we classify the animals according to anxiety using these natural exploratory tests like the open field and the elevated plasmase, then those that were highly anxious were much more vulnerable to develop stress-related behaviors, stress-related depressive behaviors. And also later on, we went through the human literature and we could see also that the knowledge was building up that those individuals that were characterized as high anxious with questionnaires like the state trait questionnaire, they also were showing higher vulnerability to develop depressive behaviors. And we wrote a couple of reviews that put together this type of concepts here. I'm going to do another kind of parenthesis within the story that I'm telling you, because I'm going to be talking about some of the research that we do in humans. And somehow this has brought me to a reflection lately that puts even more emphasis on the importance of looking at behavior. I already told you a bit that we thought maybe looking at behavior, we can reveal something. And indeed, this is the way we do it in animals. But in humans, I told you, normally what it's used in the literature is questionnaires, which are subjective, self-reported questionnaires. So what we've been developing in the lab, and this is led by João Rodrigues, postdoc in the lab, is using virtual reality to now back translate some of these approaches in animals, like for example, the open field and the elevated plasmase, what we've developed is immersive scenarios in virtual reality that I'm going to show you quickly that try to mimic the more relevant aspects of these experiences in animals, this test, and then run them in humans to see to what extent behavior in humans using this type of environments can also be indicative of stress vulnerability. And here I can run a bit. This is what somebody sees when immersed in the virtual reality. This is the empty room, like it's more or less like the open field. You can see where they start. There was the platform there with the feet. And then, yeah, the person is immersed here, moves around, has 2.5 minutes to explore as the person wants, and without any type of indication, just explore as you want around. And what we very quickly found is that if we just look at how people move in the environment without looking at any other parameter, then we could see that if we classify animals with this, the style trait test that I told you before, so that we know which ones are highly anxious and low anxious, then we see that the low anxious move quite homogeneously in the environment, but the high anxious tend to avoid at the center of the room. And then when we calculate the differences, then indeed we see that similarly to what happens in animals, that normally high anxious avoid the center of the open field, human anxiety or human anxious individuals, they also tend to avoid this. And this is very exciting because indeed we now have behavior as one way to perhaps reveal these different traits. But of course, we wanted to go a little bit beyond, because if what we are going to predict is the questionnaire, then what's the reason to use this? We want to know if behavior can be even more indicative of neurobiological or psychobiological competences or traits that could be important to understand how the brain works or vulnerabilities, etc. So instead of just focusing on that parameter, in fact, what we do is to equip subjects with sensors that allow us capture of motion and many different movements while the individuals are exploring these environments. So now I am running this one that it's like an elevated alley, and individuals can move through this alley. And this is now at the level of the high roofs of a city. It's quite stressful. I really didn't manage to go to the extent of the alley. And the subjects don't know that they are going to be put there. They are just after the exploration of the empty room, they are taken up from the platform to this environment. So with all these sensors that I was indicating, and also with goggles, we can get a lot of information about their movement related to location in the space that is labeled, and then related to the trajectories, how they move in the environment, also related to the gait, if they have a burst of gait, how is the stance, the swing, etc., and many other parameters related to the movement. And what we've been doing is applying machine learning algorithms to see if we can now be even more predictive than what we were before. Of course, something else that I'm going to tell you in a couple of minutes. But before, I told you that we were predicting trait anxiety with the time in the center only. But now with all these different parameters, the accuracy to predict trait anxiety as it's given by the questionnaire is much higher. As you can see here and here, we are now taking into account many other parameters. So indeed, behavior is telling us now a lot in defining the individuals. And as I said, can we predict stress vulnerability from behavioral data? So instead of now predicting a questionnaire, we wanted to have an index, biological index of stress vulnerability, that it's been validated in the literature, and seeing if behavior could predict the responses in this other variable. And we chose heart rate variability because it's been proved that it's a very important marker for stress vulnerability, but also for comorbidity with many other psychopathologies and also diseases. And this is a surrogate index of a cardiac vagal brake and also of autonomic nervous system flexibility. So normally we have heart rate, which is increasing when we are stressed, and then it's reflecting particularly the sympathetic activation. But when we have, or when we measure heart rate variability, it's mainly the parasympathetic input into the cardiovascular responses that it's going to be responsible, and a deficit in this control is going to be reflected by a reduction in heart rate variability. So we designed another scenario which is highly stressful. This is a dark maze that has persistent threat. Individuals are in total threat, total darkness, and they have a kind of a lantern, and they can illuminate around, but there are different items that are quite scary in the environment, and noises, etc. So the individual is tense and the threat keeps going. So stress doesn't go down as it happens in the case of these less threatening and only more arousing scenarios. And as I said before, now what we wanted to see is if behavior in these two scenarios would be able to predict the heart rate variability in the third one. And as I told you before, the heart rate variability in this third one, indeed, with time, it goes really down, indicating that indeed there is a successful induction of highly stress responses. And the model that was applied, it was a XGBoost, again with machine learning. And as you can see here, indeed, this model was able to predict at a very high level the heart rate variability. And here we have the variables that weight and account and explain this model. And again, we have that it's a number of variables. So again, behavior being in these environments a very indicative aspect. And this, I don't have time to go into details, but it's the validation of the model in other stress induction scenarios and in other, let's say, more mild scenarios. And finally here, what I wanted to show you is how high and how good this model is in predicting the heart rate variability if we compare it with simpler measurements like the time in the center of the empty room or with the other questionnaires. The anxiety questionnaire, for example, don't have a predictive value to be able to identify which individuals will have a more aberrant heart rate variability in this stressed scenario. Okay, so something else that all this is useful for now is that if we can identify a priori which individuals are more vulnerable, we can, before submitting them to stress, we can look into the neurobiological factors that could make them different and that could perhaps determine the vulnerability to stress. And in this case, now we have an anxiety test as those instruments that allow us to identify individuals that have this different vulnerability. So for this, now I go back to the rat model, but just a second, because I think I need some water. Again, we have now this model that is going to allow us to identify those animals that are high anxious versus those that are low anxious, based on the time that they spend in the open arms of the elevated plasmids, that are arms that are not protected, and normally they are anxiogenic, so that high anxious animals don't go there. And also in the open field, they avoid the center of the open field. So we classify the animals like this, and we see that when we go to the extremes, because normally the tasks that we are giving them, like a social dominance test or the force-win test, are stressful on their own, they already allow us to reveal differences in how these animals are coping in this task. They are normally showing much higher number of passive coping responses, so that we have that when we look at social dominance, high anxious animals versus a low anxious animal, the high anxious animals lose the competition, they become subordinate when they are competing. And then in the forcing test, normally they show higher levels of immobility, indicative of lack of expectations of being able to control the environment with their own behavior, even though, of course, this is a test that sometimes brings discussion as to what's the meaning and interpretation, and I will be happy to discuss with you afterwards if you want so. But before that, I want to share with you some of the recent data in the lab, looking at these individual differences, saying how do we go from trait anxiety to depression. And then, since we are focusing particularly in the motivation system, because both social dominance and coping with adversity is related to motivated behavior, we've been focusing for that reason, and also due to the results that we got when mapping brain activity in the mesolimbic system, very particularly in the nucleus accumbens, because it's involved, as you know, in motivation, the vigor of responses, effort, and also it's very much involved in the development of depressive-like behaviors in models of animal models, also in humans, and related to impaired sociability, anhedonia, anergia, low motivation, and learned helplessness. And here, when trying to relate mechanisms, it's important to be aware of the cell specificity, or the cells that are composed, that compose the ventral striatum or the nucleus accumbens. And mainly, we have 95% of medium spiny neurons, which are mainly GABAergic neurons that express different dopamine receptors, like the dopamine 1 or the dopamine 2. And then there are also GABAergic interneurons, cholinergic interneurons, and the other projection neurons, glutamatergic and GABAergic as well. And for these experiments that I want to discuss now, we've used a lot the model of social competition, because what we do is pairing the animals for equal traits and just seeing which one is going to be able to keep going and eventually win, even if they have equal chances to win. And what we could reveal, as I just indicated, is that those that are highly anxious normally have a higher vulnerability to become subordinate, to lose the social competition. And when we do these experiments, we put the animals to fight, and then afterwards look for the brain activity, which neurons get activated. We observed in the nucleus accumbens that it's mainly neurons that contain the D1 receptor, dopamine 1 receptor, that get activated, and particularly in the low anxious animals that are the ones that are winning the social competition. And we could also do experiments, pharmacological experiments, and also optogenetic experiments in which we transiently inactivated these neurons, and we could see that they play an important causal role in the outcome of the social hierarchy. So if high and low anxious animals have a different engagement of these neurons, would it be that they show differences in the structure or function? So for this, we've been doing morphological reconstructions and measuring the dendritic arborization. And as you can see, this is with Golgi staining. We have that the high anxious animals have a poorer dendritic arborization than the low anxious animals. And this is across the different types of dendrites, and this is measured as the distance from the radius. And when we look at the spine density, we also observe a lower spine density in the high anxious animals, and this is mainly reflected by a lower number of mushroom spines. We also did reconstructions after electrophysiological recordings because we could afterwards also characterize the type, the neuronal type that was recorded. And with this type of reconstructions, we could confirm that these observations that I just mentioned here, they are confirmed for the D1, dopamine 1 receptor containing neurons. And this is now the electrophysiological evidence that it's showing a lower excitatory inputs into these neurons. What we can see here is that we have the high anxious animals that have a lower engagement of these neurons, and they also have less dendritic arborization and also less connectivity. So they are less well connected with excitatory inputs coming from other brain regions, and eventually that would be activating these neurons. So why would it be? We have these animals, they are born, they both high and low anxious, they are raised in the animal house in equal conditions, but why would it be that they are different? What could we find molecular mechanisms that explain these differences? And for this, we did originally a microarrays in which we looked for differential expression between in the nucleus accumbens of high and low anxious rats. And we observed an enrichment of mitochondria metabolic genes that are different between these high and low anxious rats in the nucleus accumbens. So we decided to explore the possibility that perhaps mitochondria could be different in this nucleus and also these neurons. And this maybe could explain why there are differences. I have a few slides to motivate the importance of looking at mitochondria, even though that was not so obvious when we started doing these experiments back now perhaps 8 or 9 years ago, in which indeed it makes a lot of sense from the point of view of trying to explain mechanisms related to the engagement of neural circuits that are taking decisions regarding the engagement of the organism in exerting effort for motivated behaviors, that perhaps factors related to energy in the neurons might be different. And that's why indeed it makes a lot of sense to look at mitochondria from this point of view. And also a second reason is that we know now this is a cell, and this is cortisol, which is one of the glucocorticoids, more the type in humans. In rodents is corticosterone. We know now that glucocorticoids that can cross the blood-brain barrier and also can cross the membranes, they can control mitochondrial factors in the nucleus, but also they can activate the glucocorticoid receptor and translocate to mitochondria, affecting the functioning of the mitochondria as well, and also transcription of mitochondria and DNA. So it gives a very good substrate to mediate differences and different vulnerabilities related to stress. And I'm going to skip these slides on the sake of time, but we were wondering indeed, okay, we observed differences in gene expression, but if we look at the functioning of mitochondria, would it be that if we compare high and low anxious animals, they have differences? And for this, what we did is to take bunches of the nucleus accumbens, and then in vitro, ex vivo, we looked at the maximal respiratory capacity that different complexes of mitochondria can have when we measure oxygen consumption if we provide substrates for these complexes, so that we provide substrates to the maximal capacity so that we can see how far the mitochondria of these animals can go. And as you can see here, high anxious animals have a lower maximal capacity to keep going in these nucleus accumbens. It doesn't happen the same in all the brain regions at all. This is specific in this area. And this is something that we saw in neurons and not in glia. And it's something that we also observed at the level of a protein, not only function. In addition, because of course the electron transport chain is there and works to produce eventually ATP. When we measured ATP in these animals, they also had high anxious animals lower levels, and they had higher reactive oxygen species, higher levels of ROS. And what we also wanted to know is to what extent the function of mitochondria in the nucleus accumbens was implicated in this type of coping behaviors, and then by manipulating it pharmacologically, we saw that indeed this is involved in the outcome of the social hierarchy. So now we have indeed that these animals have differences in the dendritic structure, also in the connectivity, and differences in genes that are coding for mitochondria. And then when we look at mitochondria, there is a different capacity of mitochondria to respond. Is this important? We started looking in slices, brain slices with electrophysiology, if medium spiny neurons would be susceptible to small changes in the functioning of the complex 1. In this case, this is rotenone, which is an inhibitor of complex 1 of mitochondria, and we gave it at very low levels, otherwise eventually it may be toxic. So being very careful, and in fact, when we put it there, neurons like cholinergic neurons, they don't get much affected. These would be the red ones, the recordings. But these medium spiny neurons seem to be indeed very sensitive, and they reduce the firing frequency when the complex 1 function is reduced. Also, playing with levels of ATP, we see that these neurons are also highly sensitive to different levels of ATP in the buffer. So meaning that indeed mitochondria seems to be critical for the functioning of these neurons, at least acutely. So we wanted to get more hints because we don't see only an acute effect. When we compare high and low anxious animals, it's a trait, it's always there. Can we see something when looking at the structure of these cells that reveal differences that could be important? And when we quantify just the number or the density of mitochondria, these are electron microscopy images, and we compare high and low anxious animals, we didn't see differences, but we observed some differences. Maybe you can also appreciate the differences. We see that the low anxious animals mitochondria is a bit like smaller, and this one is bigger, the high anxious, and indeed this is capture when we measure the area and the coverage that the high anxious animals have higher, and they look a little bit like swollen mitochondria. And then we also quantify the contacts with endoplasmic reticulum, and what we observed is that they are highly reduced in the high anxious animals. They are lower levels in the high anxious animals, and this phenotype made us think, because of evidence in the literature in other cell types in the hypothalamus, that perhaps molecules that are important for fusion and also for the contact of the mitochondria and the endoplasmic reticulum, like mitofusin 2, might be differentially expressed. So we looked at different mitochondria fusion proteins or dynamics proteins. And as you can see here, it was mainly indeed this mitofusin 2, which is important for the contact with the ER, that was reduced. And as you can see here, it's reduced in the nucleus accumbens, but not in the amygdala. So because of the functions that I just indicated to you, we decided to do an experiment in which now we injected in the nucleus accumbens a virus that was going to restore the levels or overexpress the levels of mitofusin 2 in the nucleus accumbens of the high anxious rats. And then we let time for the virus to express. And afterwards, we gave a battery of behavioral tasks. And afterwards, we sacrificed the animals and measure different features of structure and function of the neurons. And first, we confirmed that indeed we restore the levels of mitofusin 2. Mitochondrial respiration from low anxious to high anxious is reduced, but this is restored in the overexpression animals. And then when looking with electron microscopy, we did a three-dimensional reconstruction of the images that we obtained. And then we got like cubes for each of the animals where we analyzed and reconstructed the mitochondria. You can see and appreciate how different shapes these mitochondria have and volumes, etc. And then when looking at, the parameters related to number and particularly surface, volume, etc., we see that the volume, as you can see, the low anxious animals are thinner. This is much more packed for the high anxious animals, and when mitofuscin 2 was overexpressed, we see a volume that is again less packed, and this is what we can see when we quantify these parameters. And when we look at behavior, that was quite striking, and it's that the high anxious animals, when we look at the percent time in the open arms, after the overexpression, they behave as the low anxious animals. And then in the force wind test, they also float less. They are less time immobile. They behave as low anxious animals, and they also win the social competition. They are not losing anymore. When we look at the dendritic arborization, the high anxious animals with the overexpression, they have a restoration of the neuronal structure, and they have a restoration of the connectivity, circuit connectivity. So now what we are trying to understand is what these differences, the variation in mitofuscin 2 expression in the mitochondria in different medium spiny neurons that project to different output regions might be contributing to which type of behaviors, and which ones can explain the differences in the social hierarchy and in the forcing test. I realized that I gave this extra or additional introduction at the beginning, and this probably is not going to allow me to finish another component that I have at the end of this talk. I'm going to summarize it without going into details. So what I presented until now here is that indeed we could identify a particular molecule from mitochondria, which is very important for how these neurons are different and how the circuit gets engaged during behavior. What we also looked in other experiments is whether these differences that we see can be programmed by early life stress. And indeed, we applied the stress during the peripubertal period, and we could see that these animals change social behaviors 2-3 months after the stress, and also their mitochondria is affected 2 or 3 months afterwards. So stress can also have a long-term impact in the circuitry. And now we have a lot of experiments in which we are looking at whether or not this phenomenon is relevant for humans, and found evidence. And particularly what we are doing is using magnetic resonance spectroscopy, which is we applied in animals and humans. And this allows us to look at metabolites in both models, and eventually see which ones are similarly different with anxiety and stress. And then what we are doing is giving nutritional supplements or treatments, and finding that this has an impact in the reversal of the behaviors, and also on the impact on stress. So I'm going to now finalize here with this image. I gave you evidence that indeed there are individual differences which are very important, and then we think that this is treat anxiety very important to define the individual. And I acknowledge the group that is wonderful. I tried to give the names while I was presenting, also the collaborators. And then I would like to thank you for your attention. This is where we're working here at the EPFL in Lausanne. And now I'm happy to answer any questions. Yes.
So thank you so much, Dr. Sandi. That was a phenomenal talk. It provided extremely important insights into the intricacies and the neurobiological basis of stress, and the experiments that are going on in your lab, especially the virtual reality ones, the open field experiments that caused quite an uproar in the audience as well. So thank you so much for that. Now we will be moving on to questions from the audience. First, I would like to ask one question that I had myself. So you mentioned that studying the genetics of this would have been quite complex because it interacts with the environment as well. But isn't stress itself the environment that we usually talk about, the gene environment?
Yes.
dialogue. So how can you differentiate?
No, I think genetics is very difficult because there are so many gene and so many gene variants that eventually one needs to have very strong statistics. And that I think the constraints related to the statistical analysis, because of so many multiple comparisons, require that the differences are huge. And this is a problem on the one hand. On the other hand, I think, yeah, I would like to emphasize a lot that indeed all we always have factors that interact with other factors. So trying to identify the weight of one gene per individual is very difficult because the gene is embedded in the genetic environment and also in the epigenetic one. So eventually, indeed, for these genes in the GWAS studies, the gene wide association studies that pop up as very high ones, are really extreme ones. It doesn't mean that all the others are not important. I think what it's important is to cluster individuals and identify that they would be important in their own genetic environment. And then related to your question, this brings a third complexity. And indeed, stress is indeed it's the environment, but also we have such different environments. How do we quantify the environment? And how do we quantify the stress? So this brings a lot of variability, which is very, very difficult then to identify. But I think progress there is wonderful. And I think we should be seeing a lot of progress. And I think now also with the stronger mathematical methods that allow us to cluster individuals, I think this would allow us to go beyond with genetics, let's say.
Agreed, totally. So we'll move on to questions from the audience now. Would Ritvika Roy like to come up and ask her question live? Ritvika? Yeah. You may unmute.
Hello.
Hello. Yes.
Hi. Hi, Dr. Sandi. It was super insightful to hear your talk.
Thank you.
I just have like a few questions, but I think the one which has been upvoted the most was my question regarding the memory formation and the rising stress levels in the brain that you mentioned at the beginning of your presentation. So, when you were conducting this experiment, did you notice any physiological changes in the occipital lobe specifically, considering that that is the key center where visual reception takes place and spatial memory formation takes place from the visual perception?
Yes. Unfortunately, the answer is no, but thank you, Ritvika, for your question, because you know how it goes in science. At that time, we were very much focusing on the hippocampus and trying to understand what could be changing in the hippocampus. And normally, at that time, we didn't think that the visual cortex was learning. This has changed progressively. So many times, it's the example for the visual cortex that you're asking. When people were seeing activity, they would just, or we would just think this is related to the visual experience, which is normal, and this is not related to learning. And I want to share with you two of these kind of thinking that it's a bit dangerous. Sometimes I think we should be more open to be surprised by what we find that come from my experience. For example, when the example that I was giving of stress or glucocorticoids, but the stress response being an integral part of the long-term memory, of the mechanisms of consolidation. When I thought about it, my supervisor at that time was establishing the criteria that one should follow to identify the mechanisms of memory consolidation. And since the brain was more seen as a kind of a computer, they indicated that anything that it's not information processing at the site of the neuron should be discarded, meaning that this was implicit that the stress and attention, arousal, emotion should be taken out, because otherwise we cannot understand how memory takes place. And I was a bit frustrated, because I thought the strength should be integral and telling the information processing that it's important there. So this is one example. The other one is with the mitochondria field that I was discussing. It's been for years and years and years that people have been collecting information regarding to gene expression, but other systems as well, and getting that mitochondria metabolism genes were always there, sometimes even the top ones, but people kept looking at them as if they were housekeeping. And this is something, it always comes, it's almost like an artifact. And I remember when, because I didn't come from this field, so I was a bit new, when I presented one of the first times, afterwards there were very, I think, respected colleagues that had been doing a lot of work with psychiatric disorders and also neurodegeneration. And they called me on the back when the session continued and said, hey, do you think what we find always these very high levels of expression of these mitochondrial metabolism genes? Could this be relevant? And I said, I think so. Oh, because we never looked into this, etc. Slowly, because the field started, I think, showing evidence, people went back also to data sets that they had been collecting. And this allows now the explosion that we are seeing, that it's placing a central role to this. In the case that you are indicating, it's very interesting, because indeed all these areas that normally were seen as sensory areas, now we see that they have also memory mechanisms that were not observed. And I think, yeah, one can revisit everything that has been done, because the models that we have is that there will be more the emotional centers or the memory processing centers, etc. But it will be very exciting, I think, to look now at the same older questions, looking at things in the way that you were suggesting.
Sure. Thank you. Thank you so much, Dr. Sandi.
Thank you.
Okay, next we have Surjadeep, who has a question for you. Surjadeep, you may unmute yourself.
Yeah, hi, ma'am.
Hello.
Ma'am, I really liked your lecture. It was so much intriguing. And my question is that from the beginning section, when the mouse was under stress, is it that much compulsory then when any people or any individual is under stress, then its memory is working more better than as usual?
Yes, of course, there are many subtleties to answer here. Indeed, it's very similar, and that's why it's good to have these models, because then we can try to understand mechanisms at another level. But of course, we have better memories when we've been stressed, but sometimes it's of some aspects. It's as if maybe stress changes the bias between the details that are kept in memory or not, because it's affecting different brain or memory systems. So sometimes we cannot only think that it's reinforcing the memory. If the stress happens at the time of the information processing, we are also processing the information differently. So, and then maybe something else to answer here is that this has advantages sometimes, because indeed we can remember some things better, but this is also related to the field of post-traumatic stress disorder, in which people that are exposed to traumas sometimes cannot remember very well the declarative aspects of the experience. Sometimes they can remember very well more the associative aspects, like more the conditioning, like a tone or something that a noise, a smell, that very quickly is going to kidnap the brain now and then bring back the traumatic memory. And that's why it's difficult for individuals to manage situations after they've been exposed to traumas, because you have sometimes an inhibition of the declarative aspects, a shutting down of more hippocampal and prefrontal cortex related events, and then a preponderance of the associative ones that are not rational and under control so much. They are more implicit memories, and that's why indeed it's what you say. It's similar to what happens in humans, but there are many different components or aspects that could be contributing. I don't know if you had something very specific in mind when you asked the question.
Not that much specific, because I couldn't understand the thing properly, as
Okay.
I'm too much novice for these things.
Mm-hmm.
So, but I understand the thing which you told, and thank you, ma'am.
Thank you.
Okay, next we have Kendresa Pardisi. I'm sorry if I'm pronouncing your name wrong. You can unmute yourself and ask.
Hi, Dr. Sandi. Thanks so much for your amazing talk. It was really interesting.
Thank you.
So I have a question concerning your measurements in the mice. I was wondering whether you look into other biomarkers as well, as you presented to us with the VR experiments. You looked into HRV measurements, for example, and I was wondering whether you are expanding the mouse analysis towards that as well, since those measurements are, of course, a bit more tangible, measuring maybe heart rates or sleep times, etc., of the mice, rather than also looking at, for example, I guess the mitochondrial activation. That's a bit more tedious, and whether there is an approach to look at those real-time biomarkers.
Thank you, Kendresa, for your question. It's very interesting and timely, because I came to give this talk to the university this afternoon because there was noise at home. And something came up to my thinking when I was parking. So I sent to myself an email to indicating that we should look at not only heart rate variability measurement, but control it in our animal experiments, because of something else, some results that I got today from the lab. So we did some experiments measuring heart rate variability and heart rate in general in animals, but it's not so easy, because of course one can do it non-invasively, but then the animal has to be mobilized, and that's not so good for the kind of time-dependent changes and more naturalistic ones. Otherwise, what we have to do is to do surgery and to put electrodes into like small veins, etc. So it's a difficult surgery. In any case, we did experiments, and indeed we can have this information. We didn't apply it to the same project that I presented here. It was a different project, but I think it would be very relevant, because something that we are doing now, I didn't have time. Also, I still don't have the results, but I show you the electron microscopy images, and this is revealing incredible aspects. Like indeed, from those images, we already can see that mitochondria is different, interactions with the endoplasmic reticulum are different. We are now looking at the location of mitochondria in different parts, and something that observing these images we see different is some aspects related to the blood-brain barrier and to the blood vessels. And this brings us back now to the cardiovascular system and your question. And that's why we think that maybe some of the alterations that we see in the brain in anxiety, as it's been also reported for stress, might be related to the blood-brain barrier. And then in this case, I thought it would be interesting to modulate the vagal control of the cardiovascular system and see if it has an impact on this. So it's a very relevant question, and I think we will be perhaps in the near future monitoring, as you were suggesting in this model, also the heart rate variability for this reason. Thank
Amazing.
You.
Yeah, that is exactly what I wanted to hear. So thanks so much.
Thank you.
Thank you, Kendresa. So next we have Sashena. You may unmute and ask, Sashena.
Hi, Sashina.
Hi, Dr. Sandi. It was an amazing talk. I wanted to ask you, did you look at mitochondrial differences between neurons and astrocytes separately in the high anxious and low anxious animals?
Thank you. Thanks for the question. We are doing this work at the moment. We see also differences, but I don't have the results to present yet. So I think it's a bit early to say much, but there are differences. Yes, it's, I think, very interesting because we thought originally that there was no reason to look at astrocytes because we didn't see differences in mitochondria respiration in astrocytes. But in fact, we know that mitochondria is not so important maybe in astrocytes, that there are other metabolic pathways which are important, but now indeed we see differences also in mitochondria, but also in other aspects of astrocytes, some expression of some key genes of astrocytic function also, they are different. So yeah.
So, so are you going to separate like neurons and astrocytes and then do like mitochondrial respiration on those, like complex activities and determining maximal respiration and all for them individually?
We already did a little bit, yes. This we already reported in the PNAS paper, and now we will do something else. We are combining now not only the rat model, because the rat model, I like it because it's a natural model of anxiety, of variation in anxiety, but in order to be very specific in the manipulation of specific genes or proteins, then we are moving to mice as well in this case to be able to do these more specific approaches in a cell-specific manner.
Yes, okay. And the other thing was that the 100 nanomolar rotenone, which you use to induce mitochondrial dysfunction. So what's the duration that it is given for if you want to induce mild mitochondrial perturbations?
Which duration for what? Sorry.
In one of your slides, you had used 100 nanomolar rotenone to induce mitochondrial dysfunction.
Yes, this was, I think, if I remember well, around 15 minutes, something like that. It's quite fast. But if you want to know precisely all the conditions, you can write me an email. I can share this with you.
Yes, okay, thank you. Because we've been thinking about this, so that's why I wanted to ask you. Yeah.
Yeah, no, but we can share this information because you know how important it is to titrate the levels, not to be toxic. So please write to me if.
Yes, okay. Thank you so much. Thank you.
You're welcome.
Okay, we have a lot of questions coming in the Q&A box, but unfortunately we're out of time. We're already 20 minutes past our schedule and we don't want to take any more of your time, Dr. Sandi. So unfortunately, we won't be able to take all of those other questions. But with this, we come to the end of the first session of Cajal Week. Don't worry, we know that you're quite a neuroscience hungry audience here. So tomorrow we shall be serving an amazing panel discussion, Bench to Bedside, Bridging the Gap in Neuroscience. And this aims to generate a meaningful dialogue between the emerging leaders in basic and clinical neuroscience. So I hope all of you will tune in for the same at 5 p.m. IST, which is 5.5 hours past GMT. Thank you again. Thank you so much, everyone, for attending.